We've been doing these public lectures for 11 years now, and I've been standing up in front of most of you for 11 years now. How many have been coming all 11 years? Not that many, okay, so that's good. Anyhow, it's nice to have you all here. We put on these public lectures every year in January, February, and March, and sometimes into April, as a way of educating the public about marine science issues and about showing what the Marine Science Institute researchers do. So most of our lectures are by our own staff and students. Occasionally we'll have visitors from the scientific community in South Texas come and speak about similar topics from their own institutions and their own institutions' perspectives. We really like doing this, and we wouldn't do it if you didn't show up. And those of you who come regularly know that, especially in January and February, we can have over 200 people in the audience. And in fact, there were a couple of times this year when we had over 260 people in the audience, which is really remarkable. And it's very rewarding to all of us because, you know, we don't get paid extra to do this, and we do it just for you. And if you didn't show up, it wouldn't be worth it. So I really appreciate you all coming out. Those of you who've come to this often know that we start out the evening by playing this week's episode of Science in the Sea. Science in the Sea is the Marine Science Institute's radio program. It's a 2-minute radio program. And the whole point of this program is much like our lecture series, is to educate the public about marine science. Now, the Science in the Sea is not always cutting-edge research. Sometimes it's things that scientists have known for a very long time, but hopefully you haven't. And so my goal has always been to have 2 responses from you. One is, that's interesting, and the other one is, I didn't know that. If I can teach you something interesting and new, we've accomplished our goal. So let's listen to tonight's episode of Science in the Sea.
Exploring Science in the Sea.
Yeah, just Victor 9, we're in Sunbury 2, 230 feet beneath the keel, transmission bird ship. Dive!
It sounds like something Q would have whipped up in a James Bond movie. A submarine bristling with lights, cameras, and a robotic arm for finding and grabbing things on the ocean floor, and even wheels for driving along the bottom. But Naval Research Vessel 1, NR-1 for short, was a real submarine designed for real Cold War missions. And when it wasn't pursuing super-secret military operations, it helped marine scientists probe the ocean depths. NR-1 was commissioned in 1969. It was much smaller than other military subs, but it could dive deeper, down to about 3,000 feet. Special sonar and other equipment allowed it to find just about anything on the ocean floor. Many of its finds are classified, but some are well known. It studied the remains of the Monitor, a Civil War ironclad warship, as well as Britannic, the sister ship of the Titanic. It recovered parts of the Space Shuttle Challenger after it exploded in 1986, and it discovered the wrecks of ancient Roman ships in the Mediterranean. NR-1 also took marine scientists on journeys of exploration. It studied everything from fish habitats to the ocean floor where new crust is forming. It measured ocean conditions and looked for evidence of ancient coastlines. NR-1 was retired at the end of 2008 after 4 decades of operations that would have amazed even Q.
Surf that.
For the University of Texas Marine Science Institute at Port Aransas, I'm Holly Brawley.
I hope you enjoyed that. And as you know, if you enjoy Science in the Sea and you don't have it on a radio station near you, just tell your radio station you want to hear it and tell them to get in touch with us. We send it to them for free. It doesn't cost them anything at all. And we'd love to get it out there. Right now, we're on 182 radio stations in, I think, 40 states, as well as a few places outside the country. So we're really happy about the program, and we just finished recording our 5th year of Science in the Sea, I think. Yes, 5th year. We just finished that 2 weeks ago. So we're really excited about that program. All right, well, without any more delay, I want to introduce to you tonight's speaker. Tonight's speaker is Geoff Hengsen, who is a graduate student here at the UT Marine Science Institute. He will be graduating with his master's degree this semester in spring, And he came to the Marine Science Institute with a strong interest in fish ecology. And so he went to work under the direction of our Associate Director for Fisheries and Mariculture, Dr. Joan Holt. Some of you may have toured the Fisheries and Mariculture Lab. I know those tours have been available recently, and I hope you enjoyed your visit there to see the big fish and maybe some of the small fish. It's a wonderful facility occupying about 10 acres of land over by the ferry landing. And some really amazing work has gone on there over many years. Geoff's work is, of course, in fish ecology, and it involved work out in the field as well. And it evolved into something known as landscape ecology, which is an emerging field looking at how organisms interact with the entire landscape, even though it's more of a seascape to speak of. And so Geoff will tell you tonight about some of the work that he did for his master's thesis. Geoff?
Thank you. How you guys doing tonight? All right, thanks for coming to my talk. Let's see, can we dim some of the lights here for this? Is that right? Okay, great. Okay, I'd like to talk to you guys about seagrass meadows here in the bays of Corpus Christi Bay and Aransas Bay. There is a great diversity in the characteristics of these seagrass meadows. And that's one thing that I'd like to talk to you about. But also, this talk is gonna be sort of a story about what it's like to do research, and really, particularly, what my story has been. I used to be in the private sector, in business. I worked for a company that produces travel and expense management software. And I was a marketing manager and a web developer for this company. And I guess a few years away from transitioning that, I decided that I wanted to act upon my love for the ocean and the marine environment, and for my questions about how it all works, and for my concern about all of the news reports that were coming in and pervading the media about the trouble that that environment was and is in many respects. And I was lucky enough to meet Joan and Scott Holt here at the Institute, and they were kind enough to give a guy like me a chance. So this is going to be a little bit of a story about what happened when I decided that I wanted to get my boots wet, because I got every bit of what I bargained for. Before I start, I'd like to also acknowledge Greg Stuntz, and Jason Williams, and Megan Robillard at the Fisheries Ecology Lab at Texas A&M Corpus Christi. This was a collaborative project between their institute and the University of Texas Marine Science Institute. And they had an integral role in administering and designing and facilitating this project and helping a guy out with his master's thesis. So, the diversity of animals and plants as we all see them on planet Earth has been something that has fascinated people for as long as we have been roaming the Earth. I mean, this is something that has perplexed and tasked ecologists for hundreds of years. Why do you see something here and not there? What you find might really depend on where you look. Presumably because maybe there are certain features about where you're looking that have some sort of a relationship with the things that are living there, that cause a group of animals of a certain kind to congregate in one area and not in another. And therefore, what you see is different. Now, in human terms, oftentimes, where you're from often depends on where you are. Now, that might be, that sounds really deep, you know, like maybe I ought to get that published on Post-it notes and make $1 million. But no, not really. I really mean this in a more simple term. And just bear with me on this example or sort of this allegory. This picture, I went on the internet and downloaded a photo from a nightclub in Washington, D.C. This is my hometown. This is a popular nightclub called Fur. And what I want you guys to do is look at all of the faces that you see in this picture. Here, 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 here. Folks, I can tell you from personal experience that when you go out on any given Thursday, Friday, or Saturday night in Washington, D.C., a city of 1 million people, these folks are from everywhere. They're from all over the world. You'll go out there and you'll meet people from North Africa. You'll meet people from Cambodia and Asia. You'll meet people from South America. A guy like me is oftentimes a minority out here at any given night on the weekend. And that's the way that this town is. It's one of the things that makes Washington, D.C. what it is. This is the city. Now, 2 miles away, there's a place called Arlington, Virginia, right across the Potomac River, okay? This photo was taken when I was approximately, 23, 25. Now, this is really a subspecies of human here. This is actually, this is a very interesting, this is Beer Guzzlus wild dingoman, okay? And including that jerk over there in a corner. And he belongs to this group of subhumans, and this group of animals congregates in the suburbs from one suburb to another and uses rooms and facilities such as this as a staging area for a weekly migration. Where? Down here. To go perform displays to various members of the opposite sex and compete with gentlemen such as this. I don't really know if he would provide us too much competition particularly, but that's what goes on. But that's not the point of what I'm trying to get across to you guys. What I'm trying to get across to you is that just 2 miles away in a suburb, I know each of these gentlemen very well. This guy is born in Turkey. He has Turkish and dual citizenship, Turkish and American citizenship. This guy was born in Vietnam. Okay. The rest of these folks are Caucasian. They grew up and spent their entire lives in Arlington, Virginia. Overall, it is a somewhat diverse group of people, but it is vastly more homogeneous than what you would find in a packed city. So there can be certain factors that are driving the mix of people that you see. And indeed, where you're from is often depends on, or who you are often depends on where you look and where you are. How about islands? I took this picture in southwestern Madagascar. I spent 2 months surveying coral reefs and working with the Vesu fishing people there. And aside from this volunteer, which was another person just like myself, out there working and surveying coral reefs, look at all of the people in this picture. In this island, just about everybody that you see here was born and raised in Madagascar and will most likely spend the rest of their lives there. Tremendously homogeneous in that respect. So again, there might be some relationships with the fact that Madagascar is this island off of Eastern Africa that might have something to do with how homogeneous this group of people is. Well, let's take another look. How about Port Aransas? Port Aransas is an island, right? And the focus of this paper, this slide it's not this beautiful sandcastle. Don't get me wrong, it's a great, great piece of art. But take a look at the people beyond it, okay? From your initial glance, it might be somewhat of a homogeneous mix of people. But I'd bet you $100 that if you went up to each and every one of them and asked them where they were from, chances are is that you'd get a really diverse mix of where they'd be from. They'd be from different regions of Texas. They might be from Louisiana, Alabama. They might be from all over the Gulf of Mexico and neighboring states. So that, you know, this island, Port Aransas, is very close to a mainland. It's connected by a causeway. You can take a ferry here. There are aspects of this island that make the people that you see here vastly different in terms of their composition compared to Madagascar. So, lo and behold, speaking of islands, there is this big theory in ecology called island biogeography. It was introduced by MacArthur and Wilson in the 1960s. And it is a way to explain or conceptualize not only islands in our most classic sense, but islands that are like remnants of habitats, a forest fragment, or a cloud forest that's on a mountain that's surrounded by, you know, desert land or farmland. It's an attempt to explain what you see in a given habitat. And there are several factors that these folks propose that have to do with it. Immigration, animals arriving on a particular island or a patch of forest. Emigration, animals leaving, birds fly away from islands and go somewhere else. Extinction, things live and die. Some species don't persist. And in addition to that, factors such as the size of the habitat, an island size, can determine how much habitat there is for various species to arrive and take hold. And its proximity to a mainland, how close you are to potential sources of new immigrants. These are things that may explain or account for what you see in a given habitat. And so, why is this important? What does this have to do with anything in terms of seagrass meadows? Well, there are seagrass meadows in both of these bays, Corpus Christi and Aransas Bay. Some of them there are the size of cars. Some of them are the size of football fields and football fields. They're huge. And it might be an appropriate framework to think of huge seagrass meadows as being cities. If you sit yourself in the middle of one, and if you are a shrimp or a fish, there are potential places to live as far as you can see. Turn around, no matter where you go. You can see potential places to put your house. And in other terms, you could put yourself on a small seagrass patch, and it could be a very, very isolated, small island place to live. It might be a framework to evaluate the characteristics of the community that you see based on these different factors. How big the island is, how far away is it from neighboring seagrass patches, so on and so forth. And this is really important because globally, and especially here in the United States, we are losing our seagrasses. This is terrible that this is happening. They are some of the most biologically productive places on planet Earth. They support huge fisheries, redfish, spotted sea trout, but they support a whole host of other animals. And biodiversity is good. Biodiversity is probability and it's possibility. And possibility is good. Currently, some of the factors that determine whether a seagrass patch in Aransas Bay is huge or if it's tiny can be accounted for by a process called habitat fragmentation. Fragmentation is the breaking apart of habitat. A big continuous expanse getting subdivided and split up into smaller and smaller and smaller pieces until they become small little fragments and remnants. That can be a natural process. It can also be something that's induced by boat propeller scarring. That's a serious issue. You can get a ticket for that around here. Worldwide, habitat fragmentation has been linked with reduced biodiversity in a number of different habitats, especially in terrestrial settings. And it can isolate populations. Sitting out there on a small island somewhere, a given resident population of animals may not be able to exchange with members of its own community coming in from other areas. And that can have deleterious effects. Studies in seagrass, as far as habitat fragmentation are concerned, have produced variable results. Sometimes diversity has been lower as a function of habitat fragmentation. Sometimes we see higher diversity in fragmented habitats. For fish and shrimp, especially in a system such as this here in Texas, we don't know. We don't know. And so I want to use this paradigm or this way of conceptualizing the various habitats that we see loosely to describe three major habitat types that we see out there. This huge continuous seagrass meadow in here. This is the size of several football fields. I'm going to liken to the big city, okay? These are actual habitats that I sampled in. Here, this one I'm going to call a suburb. It is a middle-of-the-road kind of habitat that has some areas we still have a decent amount of seagrass, but it is separated in many cases with bare sand, and there's some intermediate degree of connectivity where animals might be able to move about and exchange populations. And then finally, over down here, we've got a group of what I would consider small islands. These are highly fragmented habitats, very isolated and small seagrass patches relative to some of the other meadows that you see. And so the objectives of our study was to examine how these three types of landscape fragmentation impact the diversity of the community that you see there. Namely, what we're talking about here mostly is shrimps and fishes. My prediction was that suburb habitats with an intermediate degree of fragmentation would have the greatest animal diversity. You've got enough seagrass in there to support a healthy food chain, but there is a diversity of habitat. There's plenty of edge between the seagrass patches and the sand interface. Edges can oftentimes be very interesting places for certain animals to forage and hunt, and I've predicted that you might be able to capture some of those and actually observe the greatest levels of biodiversity. The overall design of this study was to find three examples of each of these three habitat types in a region of a given bay, okay? So that you have three examples of the big cities, three examples of the suburbs, and then three examples of the small island in a haphazard way, spread across about 5 kilometers in a given bay. We did this study in 2 bays, Corpus Christi Bay and Aransas Bay, and we examined the fragmentation and the biota, the animals living in them, in 2 seasons, summer and fall. Now, these habitats are very dynamic places. A seagrass patch is not a place that stands still. A patch can grow in very rapidly in the spring, through the summer, well into the fall, and then late fall, almost pretty much into winter, that thing can start to die back. These things change all the time, so that aerial photography or satellite photography that you take, in 2010 is not the same thing. If you actually go out there in 2011, you're going to see a completely different landscape, oftentimes. They are very, very dynamic places. So what we did is we took a GPS unit, this one, that is so accurate that when I turn it on, it will place me within about a yard accuracy, 1 meter exactly where I am on planet Earth. And you turn it on, and you walk the perimeter of this patch, and the patches inside of this plot. This plot is our focus, our camera snapshot of the habitat that is the focus of this analysis. This is how I'm going to characterize, in terms of my picture, what the suburb looks like in this case. Now, I also walked the perimeter of the entire meadow surrounding it to get the context that this suburb was placed in. Now, we're really not going to talk about exactly what these landscape features, how they're related to the biology or the diversity. We're really going to focus in this area. But my main point is that mapping and walking of these perimeters is a very time and labor-intensive process. Who here has heard of the stingray shuffle? Okay, only a couple of people. The stingray shuffle is you have to walk your way around a seagrass meadow using the stingray shuffle. You can't just go traipsing around like this. You're going to step on a stingray, and you're going to get stung, and you're going to start crying, and you're going to go home. So what you have to do, well, first of all, you have to don an appropriately soiled hat, because this is field work. And you have to turn the GPS unit on, and you have to shuffle like this. And you shuffle your way around every single patch, in every single plot, in every single landscape. Okay, for the summer, that is 9 landscapes in 2 bays. So that's 18 landscapes that you're sitting out there doing this forever, and it is a very time-intensive process. Now, first intuition about field work and research. The stingray shuffle is not as fun as the Super Bowl shuffle that was exemplified by the 1985 Bears. And because I had so much time while working and shuffling my way around the seagrass patches, I got to thinking about the 1985 Bears. And I would like to point out or ask you guys why it is like folks like this guy or any of these groups of people. I mean, it's amazing that these people could win a Super Bowl back in 1985. I mean, this is a good team. It is, but it is just amazing. I mean, these are the thoughts that you have when you're mapping this habitat after a while. It is just amazing to me. I mean, there's Jim McMahon, right? And I would really like to see what happens if they met a member of this year's winning team. I mean, the evolution of the football player is just tremendous. These are the kinds of thoughts that you have when you're doing field work at times. Yes, folks, it's not always glorious, but it is something that is a part of life. Okay. So, in the summer, this is a composite picture of the snapshots inside of each of those landscapes. That is the focus of this analysis. It's the focus of this talk. For the summer, up here we have our cities, suburbs, and islands. Examples of each, 3 examples of each in 2 bays. Okay. Sampling. Sampling these habitats involve taking what is called an epibenthic sled. It's a sled, a metal sled that has a metal box frame attached to it, and a big long net. And on the cut end of that net, you've got this collection device that captures and gulps down all of the organisms that the device has captured as you tow it and glide it over the surface of a given seagrass patch. So you pull this sled only inside of the seagrass patches. We really weren't interested in collecting animals that might have been existing here in the sand, okay? And we took approximately equal amounts of sample inside of each of these plots, whether they were in the cities, the suburbs, or the island habitats. This is what it looks like out in the field. So 1 or 2 people, depending on the design of the sled, tug this thing over the surface of the seagrass bed. And we captured 34 different species of fish, 9 different species of shrimp. We captured a whole host of crab, but I'm only going to include blue crab in my analysis that I'm going to talk about tonight, because I'm really more interested in what the animals that were associated with the water column directly over the seagrass bed were doing, and what the diversity of that community was. Oh, yeah, and we captured, collected, and identified over 522,000 animals for this study. Now, don't worry. There is this really amazing device that helps you count these animals really fast, and it's very sophisticated. You know what it is? It's a pair of tweezers. Yeah, pair of tweezers. Tweezers. That's the device. Yeah. So you take a pair of tweezers and you pick out all the seagrass out of the sample and get that out of the way. And then you pick out and separate all the fish and all the shrimp. And then, what we were dealing with were animals that ranged from a pipefish that might have been this long to a shrimp that might be maybe the size of the width of your fingernail on your pinky. And some of these animals are so small that at that stage it's very difficult to differentiate between one species and another. So what do you get to use? You get to push them all underneath a microscope. All right, so you just get in here and do this for hundreds and thousands of animals. And we also measured a subsample of every single species in every single sample. So that we have size and approximation of the size or the life stage of these animals as well. So I want to say that, maybe I'm going to ask you guys, how many of you guys have heard the expression that old fishermen never die, they just smell that way? Right? Okay, well, it sounds like a lot of people have. I'll be the first to admit to you guys, I am not much of a fisherman, but after digging my hands through dead fish and dead shrimp and dead crabs and ethanol and formalin, when I came home from the lab every night, at least in my wife's eyes, I smelled like a 150-year-old grand champion of the deep sea roundup. Okay? That is lab work. That is part of the research experience. And I think it's a very important one. So first of all, let's go ahead and see what we got. And the first thing I want to talk about is just animal abundance. Let's just set aside species, the number of species and diversity, and just say, hey, is there a difference in the amount of animals that you find in cities, suburbs, or islands? Okay, this graph here compares these 3 habitats. So we've got big cities here, suburbs here, and islands here. In red is what we found in the summer, and in blue is what we found in the fall. And so what this is animal density, the number of animals that you will find in 1 square meter of seagrass. And the first thing that I want to point out to you is that in the summer, there's a lot more animals, period, big time, okay? The second thing that I want to point out to you is that in the summer, it's actually the island and suburb habitats that are totally packed with animals. The big cities, the big continuous seagrass expanses, per square meter, they don't have as many animals. It's almost as if, it's like my analogy of, when you think of big cities, in human terms, that's where everybody bunches together. Well, in the big cities of seagrasses are places where there is room to roam. And so it's almost as if those animals just don't feel and act as crowded in the summer. In the fall, however, you see an almost opposite pattern. In fact, there are greater densities of animals in those big cities. And that's very interesting, at least in my mind. And I'm not 100% sure about what is going on there, but it might have to do with the fact that in the summer, we're not experiencing huge cold fronts like we are in the fall. We're not experiencing the dieback of seagrasses that might really impact these highly fragmented areas first. But this is really important. Why? Because animal abundance may influence how many species you see. It may bias your observations or your measurements of diversity. Let me give you an example. I could tow and get 3 meters of seagrass sampled out in the meadows and put 100 small animals in this vial, this little guy right here, okay? I could do the same thing, go somewhere else, tow 3 meters, and maybe I might find 1,000 animals and put them in this jar. 100 animals in this one, let's say I find 5 species. 1,000 animals in this one, and I find 10 species. I sampled the same amount of seagrass, so in one sense, I might say this is the more diverse assemblage. This is the more diverse animal community. But is that true? It might be that there are so many more animals packed in here that my chances of encountering more species are far greater. That if you actually somehow magically use probability theory to extract 100 animals out of this sample here, and you only found 2 or 3 species compared to the 5 here, this one would actually be more diverse. So, let's talk about diversity. And first, let's look at things as if we're setting the density issue aside and just say, look, in sampling equal amounts of seagrass in this plot or this habitat or that one, what do you get? Okay? All things equal and saying that you're looking at samples that take in equal amounts, square meters of seagrass sampled. First of all, in the summer, diversity, this is the number of species here on this axis. Here are our 3 communities, big city, suburb, and island. In the suburbs, in the summer, overall diversity is greater than in the fall. When going out there and just sampling per square meter of seagrass. Okay? The second thing is that there are, on average, a greater number of animals in the highly fragmented habitats than in the continuous big city communities. That's quite surprising. In the fall, almost practically, at least in statistical terms, there's no significant difference between these. There might be slight evidence for some sort of trend if you were to sample more to see an effect. But really, in statistical terms, that's pretty flat. Okay, so there are different relationships here when you're comparing the number of species per meter squared of seagrass that you sample. What happens when you standardize your sampling effort on equal numbers of animals that you were to extract out of a given habitat? Here are the numbers of species. What I did is I programmed a computer to randomly sample 865 animals out of every single habitat using probability theory. And what do you see? Well, first of all, there's no significant difference in diversity per numbers of animals between summer and fall. There's no significant difference on average between overall what you see in summer and overall what you see in fall. That's interesting. Secondly, although there don't appear to be statistical differences between the 3 habitat types here in the summer or between these 3 habitat types in the fall, there is a slight opposite trend that may exist out there. Perhaps if you were to look at the data in a different way, that you actually might find greater diversity in the big city continuous seagrass meadows than the highly fragmented ones in the summer, and then an opposite trend in the fall. So when you ask the question of biodiversity, it really can depend on how you look at it in terms of the amount of area that you see or sample, or the number of animals that you count, because differences in abundance can bias your picture. Typically, in a lot of research, a lot of researchers are presenting both ways these days. I would like to talk a little bit about another way of looking at diversity, and that is something called species evenness. Let's set the number of species aside. Species evenness is a measure that gets at how evenly or equally represented various members of a community are. So if I had a basket of fruit, and there was 30 pieces of fruit in there, 10 bananas, 10 oranges, 10 apples. That's a very equal representation of these three types of fruit. It's very even, and I would say that that's quite diverse. But if I had another basket, 30 pieces of fruit, and I had 29 bananas and 1 orange, that's really uneven, okay? That, in terms of species evenness, is far less diverse than the basket here in my right hand. And so the next graph that I'd like to put up here is measuring species evenness and comparing those three habitat types. Again, we have here the islands, the suburbs, and the big cities. And what do we see is that regardless of whether it is summer or fall, species evenness, the animals in the big city continuous seagrass meadows are more equally represented, the species there. Whereas down here in the island habitats, there are certain members or one member of the community that is absolutely dominating the assemblage. It is making things very uneven. And the suburbs are in the middle of the road. We see a clear trend across these three habitat types. And furthermore, there really aren't any significant differences seasonally. Even though these red bars are somewhat higher than the blue ones, statistically speaking, the patterns of evenness when you compare one season to another are not statistically different. But the patterns between these habitats are very clear. So that is also very interesting. So let's talk about the patterns or the different, how these different species are responding to these various lands, these three categories of landscapes, and what is driving the patterns in evenness that I just displayed. And let's talk about summer first. First of all, what I'd like to present to you here is a comparison of relative animal abundances. All you guys really need to know is that these numbers down here are just codes that represent individual species. Doesn't matter exactly who they are at this point. They are just saying, you know, this is species 1, species 2, and this is a plot of their relative abundance. Over here on the y-axis, this is these values have been log transformed. What does that really mean? What that means is that the difference in the height between this bar and this bar is huge. I had to do this in order to make this graph presentable, you know, without our eyeballs going all over the place, okay? The difference in abundance, relative abundance between this species and this species is absolutely ginormous. Why is that important? Because this community that I've displayed here comes from the isolated island patches, the places that were very uneven. Who was dominating this assemblage? These guys were. Arrow shrimp. These are absolutely piled into these highly fragmented small patches, and they got the run of the place. Now, don't get me wrong. Look at all of the other species that are present here. It's, as we've seen in the other graphs, it's a diverse place. I mean, in terms of the raw number of species. But it's these guys that are running around having a party, okay? Let's compare it to the big city. We see a very different pattern, a very different pattern. All of a sudden, the arrow shrimp right here are much lower down. They're not as dominant, not nearly as dominant as compared to a number of other species. Who are they? Well, we've got a number of grass shrimp here, Paleomonites vulgaris. This is not a true grass shrimp. This is Hippolyte plurocantha. Ooh, Penaeid shrimp, kind you eat. They're up here. They're making a move. Gobies, darter gobies, code gobies, naked gobies, blue crab. All of these animals, in terms of their relative abundance, are really starting to assert themselves. So it's a much more even place in terms of the representation of different species in a given habitat. Still very diverse. So if you overlay the graphs between these three communities together, you can kind of see where those animals are making pushes, okay? A couple things that I think are kind of interesting. I've got a list of some really rare species. This isn't something, they're so rare that you can't make any definitive conclusions about an individual animal per se, about how it responds to a given habitat or why it's in highly fragmented areas, the island communities versus the cities or the suburbs. But I've put blue squares next to the species where they were only found in that specific habitat. For example, bay anchovy, bluefish, spadefish, only found in the isolated islands, okay? I think that's interesting. Why? Because, first of all, these are really crazy fish to be finding back there in the seagrass beds. It's a very rare thing. These are animals that are typically associated with the water column, that are not particularly associated with seagrass beds. But hey, maybe these highly fragmented, isolated patches, they have so much edge and sand interface to them, maybe the probability of an animal wandering around in the water column is greater for encountering those habitats than, say, in a big continuous seagrass bed that I've termed a city. Maybe that's why we're finding them there. So there might be something to say collectively about those animals. Not a whole lot of things that I can really draw in terms of conclusions about why you find a hog choker or a sheep's head, or a frillfin goby or a baywhiff here in the suburbs. I mean, the baywhiff, your goby, your hog choker, those are pretty benthic, bottom-dwelling animals. Your baywhiff's a flatfish, like a flounder. Okay, I'm not really sure why I only found examples of those in the suburbs, or why I only found killifish and sheep's head in the cities. Can't really come to any good ideas as to why that might be. Maybe you guys might. Let's take a look at things in the fall. Oh, before we talk about the animals in the fall, we have to talk about something else. This was another hallmark of a research experience. This is true about doing scientific research. The unexpected happens. So, these seagrass beds grew from the time that I mapped them in the summer. A few months later, when I went out there in the fall, things had changed tremendously. This is an example of a summer habitat, and I've laid under what happened in terms of the seagrass growth in the fall. Let's try that again. Over here, summer habitat. Oops. Oops, I don't know why that came by. Looks like that one layer didn't come through. But some of these seagrass beds, especially in the suburbs, had grown in so much that they turned into cities. I had to select new habitats. So what did this precipitate? This was the phone call. This is me. I'm out there. It's late August, September, and I call my wife. Hello? Hey, baby, how you doing? You know, whenever you say that to me, that usually means you're about to drop some bad news on me, and it's probably something that you did. Yeah, feminine intuition. Yeah, honey, I'm not going to be home for dinner tonight. As a matter of fact, there's going to be a lot of nights that I'm not going to be home for dinner. Why? Well, I just found out that all the seagrass patches that I mapped during the summer, they all grew in. And I'm going to have to remap every single patch in every single plot, in every single landscape in both bays. Honey, are you there? Yeah, you know, Geoff, how long is it going to take you to do this master's thesis? Baby, when we talked about getting serious and moving in together, I told you I was going to go do this master's thesis, and there'd probably be days like this. That is a reality of research too. So that is something that I'd like to point out, in that here are the snapshots of the landscapes between summer on the left and the fall on the right. And especially in our suburb type habitats, you can see the degree of growth that actually happened. The same thing here as well in the island communities. So this was very important to remap these habitats in order to accurately capture the relationships between what the landscape is telling you and what the biota inside of the landscape is trying to tell you, and how accurate your assessment of those relationships are. Now recall that I had mapped areas that extended way beyond this, so that in the end of the day, between summer and the fall, we had mapped 72 football fields of seagrass meadows to sub-meter accuracy. So that was the end of the day result. So let's talk about the biota in the fall. Same pattern that we see here. Domination by Mr. Arrow Shrimp up here in the highly fragmented habitats, our islands. And then I'd also like to point out a very important arrival in the fall, our red drum. Having been spawned offshore, these animals are being advected and transported in via wind and tidal dynamics, and arriving and settling on the seagrass beds to grow there as young juveniles. Spotted sea trout are a prominent member here, although we did definitely have those in the summer assemblages. These animals are there all the time, as opposed to their cyanid relative, the red drum. Fall cities, a similar shift in evenness. So we're seeing a number of the similar animals making a push up in terms of their relative abundance. We still got a good push here in the gobies and here in our grass shrimps. Pipefish really made a big climb here as well. There are 3 different species of pipefish out there: the dusky, the gulf, and the chain pipefish as well. So a very similar pattern in terms of the shift in evenness and the equality of representation between fragmented habitats versus big continuous ones. And so you can kind of see that when you overlay these graphs on top of each other. Okay, so thus far we have considered these habitats in terms of cities, suburbs, and islands, 3 categories. But I want to invite you guys to consider these habitats in terms of a gradient. Gradients exist out in nature as well. That maybe instead of thinking about them in terms of 3 different categories, you think about cities that are of varying sizes, suburbs, which might be more fragmented or less fragmented, islands, some of them are bigger, some of them are smaller, some of them are closer together, some of them are farther apart. There's a gradient that exists out there. If you walk your way through, you might actually notice that as you go from a very continuous expanse to increasingly more and more fragmented landscape. And that in terms of analyzing the relationship between habitats and the biota, you can do the same thing. And a very interesting result can occur. Recall that this is the graph where I had standardized the diversity in terms of the number of animals that you see. We didn't find any significant differences between these 3 categories, either in summer or fall. This is a graph that we have already seen. But what happens if you analyze them in terms of a gradient? Okay, down here on this x-axis, we go from highly fragmented habitats to ever more increasingly less fragmented, continuous, really, really big city over here, really fragmented, isolated islands over here, okay? Diversity, low diversity down here, very high diversity up here. And what do you see? Well, in the summertime, you see a very striking pattern that species richness increases significantly as you go from highly fragmented habitats into the more continuous habitats. Okay, to put it another way, as fragmentation increases, biodiversity in terms of number of species, standardized by the number of animals you see, decreases. And so that pattern really pops out. Now the slopes of these lines here kind of look different. You know, I've got them comparing. The blue ones here are Aransas Bay and Corpus Christi Bay. Here's here in the red. But statistically speaking, they're not, those slopes of those lines are not significantly different. That if you look at this collection of X's and O's, there is a highly significant change as you go from very diverse, continuous communities down to the highly fragmented ones. In the fall, a very interesting pattern appears. Corpus Christi Bay, okay, we've got very highly fragmented habitats over here, going to very unfragmented, very continuous places here. All of a sudden, species diversity is highest in the highly fragmented areas, and it decreases very rapidly in the continuous. Aransas Bay, geographically speaking, that relationship did not appear. Okay, this line may be put in there by the computer program, but statistically speaking, the model is very unsure of itself. Okay, it is really communicating to me that this cloud of points might be kind of like that, but statistically speaking, there's so much variability in there, I am very uncertain. And that statistically speaking, I can't find a relationship between habitat fragmentation and diversity and number of species in this bay. But I hope you guys can see that pattern was very clear here. So that brings in this whole other aspect that, okay, we've seen some very interesting differences in terms of what happened in summer and potentially in fall, but there can be geographic differences too. This might be a very unstable relationship. So, conclusions. Restricted, isolated habitats can have very different diversities in terms of species richness and species evenness than continuous expanses. Those patterns can depend on whether you're looking at them in the summer or the fall, and it can also depend on how you want to analyze this data and look at these habitats in terms of discrete categories versus a continuous gradient. Really, what I want you guys to take home from all of this is that diversity can appear in all of these different habitat categories or across the gradients, and that conservation of all of these habitat types in the seagrass meadows is vitally, vitally important. And so I think that that's something that conservation managers really need to keep in mind. And with that, I would like to give special thanks to Joan and Scott Holt for giving me a chance to explore these seagrass meadows and work on my master's, and other members of my thesis committee. There are a wonderful group of very hardworking professionals here at the Institute and graduate students as well that I would like to acknowledge. My beautiful wife, Alyssa, and Ken Dunton and his lab for his facilities and using GIS resources to look at this stuff, and Emily Williamson for helping me do the species ID. And with that, I'd like to thank you guys once again and take any questions. So the question, and this is a very good question, the question is whether or not, or what I think you're getting at, is whether or not the process of fragmentation and the response by the biota to fragmentation is something that is stable and observable over time. This is very, very important because this study looks at snapshots of landscapes and how they evidence themselves, the patterns that you see and the animals as they were at that one particular time. And we did this in 2 seasons. But as you guys have seen, these habitats are very dynamic, and likely so is the response of the biology, of the biota that's going on in there. And I think that it is very, very important to study the process of habitat fragmentation over time, as you're suggesting, as opposed to the end result or the snapshot in time of what the biota did as a function of the end result of that process of fragmentation. This is a very, very hard thing to do. It is something that is time and cost intensive, but is very well worth doing. There are very few studies that look at the process of fragmentation because in many instances that involves ripping up seagrass, which is practically illegal anywhere seagrass is. So it's really hard to do. I will say this. There are a couple studies that have shown that it's really more telling to look at the end result of what you see in there in terms of what the biology is telling you at the end result and that snapshot of landscape pattern. That patterns emerge there, whereas some studies that have actually gone through the trouble or gotten the permission to disturb and rip up habitats oftentimes don't find statistically significant differences in what you see there. However, that can be very system and very species specific. So the work definitely needs to be done. Sir?
What about the weather, the storms that come up? Doesn't that tear up some of this stuff and change things?
Absolutely. Absolutely. The question is how does weather impact these habitats? First of all, sir, I'd like to point out that we went out there and you sample these seagrass beds in one week, and you go back there 10 days later and you sample them again, just to get a better picture of what's going on. One of these events in the fall came back really weird. Up in Aransas Bay in the fall, densities and the numbers of species went and all I can tell you is that between the first sampling event and the second sampling event, I'm pretty darn sure we had a pretty nasty cold front that came through. That can definitely affect the biota, but it can also affect the habitat. I mean, it is, after all, this is a natural form of habitat fragmentation. This stuff is being mitigated by how the seagrass grows, but the seagrass grows and dies back in response to environmental variables. Wind energy, wave energy, the amount of sunlight, nutrients, all of these things are coming together and creating this symphony that is expressed in the landscape pattern. So it's very important to do. And I also would venture to say that it would be very interesting to see how the variation in tide influence these animals. That maybe, you know, as the tide goes out, they might pile onto various islands or ship off and go into deeper water. that would also be a very interesting environmental variable that could come into play. Sir?
When was these samplings done in relationship to this oil spill problem? Do you think that would have an effect on water?
So the question is did the oil spill influence our data? The short answer is no. We were fortunate enough to have conducted this study before all of that tragedy and mess happened. What I will say is that in the fall, we had a red tide that occurred in certain portions of the bay. That was recorded, and there were some fish kills associated with that. And we were very worried about an event like that happening, and that I would conjecture to say that an oil spill could, if it were to occur in the fall, that could have terrible, terrible implications for our fall collections. Why? Because if an oil spill happened off the coast and there's all this oil in the water while the red drum are spawning, I mean, all the dispersants and the oil itself could have terrible effects on larval fish as they're being spawned and making their way in through the fish passes and arriving on the seagrass beds. I mean, I would imagine that an impoverishment of the planktonic environment could happen, and there could be subsequent impacts in terms of reduced food for the various shrimps and fishes that are feeding off the water column to some degree, and maybe feeding off of animals that are more associated with the seagrass beds themselves. And God forbid if that stuff was actually just to sock the seagrass beds, it would be horrible. Sir?
Did you attempt any other correlations? You've talked about a number of variables and things that could impact your findings. And of course, this leads me to conclude that you possibly look at other ways of presenting that data. And what have other people done? How does your work compare to what others have done?
One of the biggest things, or the biggest regrets that I had, is that I didn't have a clone of myself for this study. Because if I did, I would tell that guy to go out and shoot densities, take cores of every single patch and multiple points inside of every single patch that we sampled. Why? Because not only do these seagrass patches vary in terms of their size and spatial configuration, so on and so forth, but the actual densities of the shoots can vary in these seagrass patches. That's something that has been studied, and there have been differences in the diversity of animals that you see in very structurally complex seagrass meadows versus ones that are more sparse. The growth and dieback that occurred in the fall may have influenced the animal diversity of the assemblages there. When you go out there in the fall, the shoot density is far less than it was in the summer. That would have been something I would have loved to have done and incorporated into some sort of a stepwise multiple linear regression to say, well, how much of the diversity is accounted for by habitat amount? Okay, well, how much of it is by structural complexity? And there's even ways that you can partition the variance and the variability in diversity due to these various factors. That's one thing. My models, some of the best strengths that I've had is something about 60% of the variability in diversity can be described by aspects of habitat amount, connectivity, and proximity. What about that 40%? I mean, there's so much variability out there. There could be temperature, water depth, salinity, things like that. I mean, we measured those things when we sampled, and I didn't find any really, really convincing correlations between them. But there are undoubtedly other things out there that I would like to get to. Yes, sir.
In your sampling, you have three of each type of community in 2 bays, three types of communities. How did you decide that that's a large enough sample to draw conclusions from?
Okay, well, ultimately that really rests on the type of statistical design that you're actually going to employ. if you want to analyze this data in terms of assigning the data points to 3 explicit categories, you are going to wind up with a design if you have 3 examples of each of 3 types and do an analysis of variance. Probably the power of your test is going to be a little low. What we decided to do was sample really intensively to try to reduce the variability that you actually get or in those samples, or increase the amount or the power of your test to account for that variability. If you analyze it in terms of 3 categories, you saw that the error bars were pretty high, you know, and that in terms of the species difference, if you're really only looking at a difference of maybe 2, 3, 4 species, the variability between those 3 categories is not enough for your statistical report to say, that's a difference. But if you use more linear regression-friendly methods that really plot you know, these plots along an ever so more so increasing gradient and look at the diversity there, the way that statistical model handles the variability seems to have come upon or detected a relationship between those variables in at least in my humble opinion, a little bit more clearly. So it's also, to answer your question directly of how do you determine the number of replicates and so on and so forth, some of it is convention based on other studies. There are oftentimes people who have previous data that says, well, in our other studies, when we sample this much, you usually get 22 species, and sampling, you know, a whole lot more or including a whole lot more replicates doesn't really get you all that much. You know, you can draft species accumulation curves that shows how many species you get as you progressively sample more and more and more habitat. And what my last analysis does is an analysis based on species accumulation curves. Because out there, frankly, sir, you're never going to hit the limit of the species that you're going to discover out there. The curve will always increase. Even if it flattens out, it will always increase more and more and more. But there are statistical procedures such as rarefaction that just compares how those curves are going and compares the heights of those curves or specific values, which is what that analysis actually did. Yes, sir.
Have there been other studies similar to the one that you've done in other areas along the Gulf?
Yes.
And how does your data compare to those data?
Well, first of all, at least in terms of biodiversity, it's difficult to compare this data set because we were using an epibenthic sled that was really more geared towards looking at what's going on in the water column. There is another data set from Johnson and Heck over from the University of Alabama, or the Dauphin Island lab there, that looked at diversity by taking these big traps and just dunking them right on seagrass beds and then suction sampling all the crabs and all the shrimp and fish out of there. And their study was really more inconclusive. Too much variability out there. It was really hard for them to come up with a clear and significant pattern across seasons and even across habitat types within a bay. Yes, ma'am.
You talk about the sled that you drag across there, and then you say you don't disturb the grass. How can you do both?
This is a very, very good question. The question is does our sampling in the habitats itself disturb the community and potentially systematically bias our data set? On the count of, going in there and sampling 1 week with the sled, and you're zooming over it, and then, you go back, 10 days later, presumably after the animals have settled down, reorganized, and you do it again. Well, we may have defaunated those patches by sampling 1 week and going back 10 days later. I performed a repeated measures analysis of animal densities and diversities comparing patch by patch, plot by plot, what happened in sampling event 1 versus sampling event 2. And the only thing that I could really conclude was that there weren't any systematic biases that that sampling might have introduced or done to the data in there. I think that that kind of sampling does disturb those habitats to some degree. It may have added some variability to the data model. However, at least in terms of the use of rarefaction and the drafting of species accumulation curves, I used a type of rarefaction called individual-based rarefaction, at least for tonight's analysis, which really ignores the sequence in which those samples and animals comes in. I mean, it satisfies the design or the use of that type of method in terms of the fact that we went out there and got samples from multiple toes in multiple different places, but any systematic bias in there is not accounted for. I have prepared and set up an analysis that uses rarefaction based on the samples themselves, where potentially a bias like that might occur, but we are pooling the toe transects within each of these environments. And so unless I have differentially affected these habitats in some way, which my repeated measures analysis doesn't suggest that I did, I'm hoping that I'm okay. Yes, ma'am.
My question wasn't that good. What I really wanted to know was,
Oh.
did you disturb the seagrass? When you take a
Oh,
sled,
you take a small chunk of seagrass out along with the biota in there. It's mostly stuff that's really, really loose and just hanging around. It doesn't. You don't. When you pull that seagrass or that sled through the seagrass, you don't see like this bare spot or anything of that nature. No, I mean we would never be issued a permit to do this kind of work. Doing that, never. Yes, sir.
One more. What further research do you think that you're spending calls for?
Personally, if I want to do a PhD, and you know what I want to do? This is what I want to do. I want to take our GIS photography and satellite imagery over the past 20 years, and I want to develop a model, a computer model that looks at how that seagrass has changed in Corpus Christi Bay and Aransas Bay, and try to develop something that explores and looks for correlations between key environmental variables, environmental forcing, severe weather, salinity changes, turbidity, nutrient dynamics, puts that stuff together, go back out there, and over the course of a year, map those habitats again and again and again and again, sample those habitats again and again and again and again, fall, summer, spring, winter, and really get a very progressive picture in situ of not only how those seagrass habitats are changing in response to the environmental variables, but the biology as well, looking at how they're responding to meteorological events as they're impacting and influencing the configuration of those seagrass beds. And if my field data agrees with the larger, more long-term data from a 20-year data set of seagrass growth, try to come up with a comprehensive picture as to how climate change, as predicted in the IPCC reports, there's 4 different scenarios, what is going to happen in the next 100 years for the biodiversity of seagrass beds, and what are the implications for shrimp, for fish? What are the implications for animals that are born in seagrass beds and stay in seagrass beds? What is the implication for fish that are born offshore and land on seagrass beds? Completely different dispersal strategies and life cycles. What's the implication? Climate change, seagrass loss, and organization and reorganization of biology. We have to get there.
Okay, well, let's thank Geoff for his presentation. Let me just put in a plug for the last two talks of the season. Next week and then the week after that, we have guests from outside the University of Texas. We have, in addition to our normal public lecture series, we have an endowed lecture series called the Laura Randall Schweppe Endowed Lecture Series in Marine Science. And we use that series, it's funded by an endowment, as the name implies, to bring in accomplished scientists from all over the world. And we usually bring about 3 or 4 of these people in a year. And the next 2 speakers are guests of that lecture series. Next week, on Thursday, is Dr. Karin Limburg, who is from the State University of New York at Syracuse University. She's coming down here during their spring break, like so many others. And she's going to work with our scientists and our graduate students while she's here, give us a technical seminar, and give a public lecture next Thursday night on ear bones and dead zones. What can fish otoliths tell us about hypoxia? And I'll let you come to learn more about that. And then the following week on Tuesday instead of Thursday, we'll have another lecturer from another place coming for the Schweppe series, and that'll be Dr. Stephen O'Brien from the National Cancer Institute, a very distinguished scientist. He'll be talking on the subject, Tears of the Cheetah, Roar of the Lion, Uncommon Glimpses into Survival in the Wild. I think you'll enjoy both of those, and I hope you can make it. Thank you all for coming, and please be safe going home.